Going to post this link. So today, the links that I just shared are the slide decks that we're going to be covering today, and in order. So the very first one is the DNA slide deck, which we're going to get into. Then the next one is DNA technologies and DNA manipulation. And then the last one is where I'm hoping to get to, and that is lipids. Um, but with all that in mind, I'm going to go ahead and launch a couple of poll questions for you guys. These are just kind of like refreshers or reminders or, things to think about with respect to last lecture. And we're polls. There we are. Um, so I've got DNA, RNA starter questions. There's a total of three questions here, so go ahead and take a moment and answer those. While you're doing that, I'm going to go ahead and get full screen action on DNA. Go there. Full screen nucleotide structure. So I've got the poll question. Okay, great. Got one person who's answered them. Three, great, four, and so on. And I'm going to give you, we've got plenty of poll questions today, so go ahead and answer these. If you don't get to these, there's going to be plenty more. What we talked about last time was we were talking about DNA and we were talking about some of the components of a DNA molecule and getting into some of the terminology. So there's this term nucleotide, and then there's a similar term, nucleoside. Some of you are familiar with this. The first poll question really gets after the difference between, or what a nucleotide is. A nucleotide is composed of a nitrogenous base, a sugar. So here's our nitrogenous base, here's a sugar, and at least one phosphate group. So the correct answer for that is C. Now, if a structure contains ribose and adenine, but no phosphate, it is a nucleoside. That was our second question. And then the last one is what is missing in deoxyribose compared to ribose? Okay, so if you look at this sugar, we've got a ribose molecule. And we know that it's ribose because this position right here has an OH group. And everyone is correct in their selection of, or the majority of the class is correct in selecting C, 2' OH. Okay, so like I said, there's going to be plenty more poll questions. Now, next up, so I'll share the results. Sugar plus base plus phosphate is a nucleotide. If you have ribose, a sugar, and adenine, a nitrogenous base, then you've got a nucleoside. And then last but not least, what is missing in deoxyribose compared to ribose, and that is a 2' OH group. Okay, if some of that was a little bit like, what, huh, where? We're gonna get through that. Okay, now that's not what I'm looking for. Instead, oh, there we are. Um, so that's where we ended. Or this is the slide that we ended on yesterday, where this is just trying to introduce some of the components of a nucleotide. So a nucleotide is a nitrogenous base, a sugar, and at least one phosphate. If you have multiple phosphate groups, well, that would be a monophosphate, a diphosphate, or a triphosphate. Now, those phosphates, the farthest one out, the farthest from the sugar is known as your gamma phosphate. The second one is the beta phosphate, and the first one coming off of the carbon on our sugar is known as the alpha phosphate. So alpha, beta, and gamma. Now, there are some subtle differences that need to be recognized. If you ever talk about a molecule of ATP, ATP is different from DATP. This lowercase d right here indicates that you are talking about a deoxynucleotide triphosphate. So what that means is we're talking about a molecule that does not have an OH on this position right here, that 2' O or that 2' OH is missing. So D stands for deoxy. So I'll just write that as no OH at the 2' position. Now, whenever we talk about nitrogenous bases, the nitrogenous bases that are found in DNA and RNA molecules are shown here. Now, the first two molecules that are shown, or the first two structures that are shown, are that of a pyrimidine and a purine. Now, a pyrimidine is a single ringed molecule. It is a aromatic compound. A purine is also an aromatic compound. Now, if you think about aromatic compounds and you think like, okay, well, there's alternating double bonds or delocalized electrons. Those are things that come to my mind whenever I think of aromatic compounds. But one other thing that comes to my mind is, well, those aromatic amino acids are how we detect the presence of protein in a sample. Same thing goes for, well, these aromatic compounds right here, purine and pyrimidines. Those delocalized electrons allow us to use the Lambert-Beer law to detect and quantify DNA. Now, what I want you to know about this is I want you to know which bases are which. So adenine and guanine are two bases that are found in DNA that are examples of purines. So purine is kind of your umbrella molecule topic or grouping, and then adenine and guanine are examples of them. Now, what I want you to recognize also is the numbering scheme. So when it comes to numbering of atoms within the rings of purines and pyrimidines, for purines, we start here. 1, 2, 3, 4, 5, 6, 7, 8, 9. We're going to the larger of the two rings, and we're going in a counterclockwise fashion from the nitrogen that is our highest priority. Now, I bring this up because if you elect to pursue some sort of cancer biology, or if you become a physician and you're studying, if you're an oncologist or something like that, um, lots of cancer therapies are modified forms of nitrogenous bases. For instance, if you had this right here, let's say we took this guanine and we methylated it. By that, I mean we added a methyl group to nitrogen 7. This would be an N7 methyl guanine. Now, this is just a hypothetical. I don't think that N7 methyl guanine is an example of a cancer therapy. However, I'm just using this as a model. A methyl group, I don't know that that's going to change anything. A methyl group for a acetyl group or something else, some sort of chemical modification to a nitrogenous base could be administered as a drug. And essentially, that drug would maybe disrupt part of the cell cycle. So it could be problematic for the cell and those proliferating cancer cells. Okay, so your purines are adenine and guanine. The numbering, I don't need you to know the numbering. I don't want you to stress about the numbering, but what I do want you to recognize is that the numbering that's reserved for the atoms within your bases are simply 1, 2, 3, 4. It's not 1 prime, 2 prime, 3 prime, 4 prime, and so on. It's just non-primed numbers. Okay, the other class of compounds are our pyrimidines. We've got cytosine and thymine. These are two pyrimidines that are found in DNA. Cytosine and uracil are two pyrimidines that are found uniquely in RNA molecules. If you look at these structures, the difference between thymine and uracil, well, it's just the methyl group. Or it's pretty much just that methyl group right there. Your pyrimidines are also fairly simple. They don't have a lot of modifications. The numbering all starts at this nitrogen 1 position. Again, 1, 2, 3, 4, 5, 6. That's how you number the atoms within a pyrimidine ring. We're not looking at 1 prime or 2 prime or anything like that. Okay. Now, when it comes to nomenclature and important rules of nomenclature, just as sort of a summary of what I just said, was the primed numbers. 2 prime, for instance, references atoms within the sugar. That sugar is either ribose or deoxyribose. Your non-prime numbers are referencing atoms within your nitrogenous bases. So 2, 3, 4, and so on. Those are in reference to the atoms within the rings of your nitrogenous bases. The phosphate group may be attached to the sugar at one of two positions. It could be at the C3 prime, in which case you have made a 3 prime nucleotide. Now, if I jump back for a second, could anyone tell me how would you refer to this molecule right here when it comes to a nucleotide? Is it a thinking about the numbering of the carbons in that atom, or the numbering of the carbons within that ring? Try to integrate the prime numbering and, well, what is this whole structure right here? Not worried about the base, done. Not worried about this being a deoxy or a ribose. This is a ribose. But how would you classify this as a nucleotide? Add it to the chat. Or how would you refer to this as a nucleotide? Well, we know that it is a nucleotide for starters, because we've got at least one phosphate. We've got three phosphates, so we could refer to this as a triphosphate. Okay. But the question that I was really asking there was, well, what is it? I'm referencing this carbon right here. Yeah, it's a nucleotide triphosphate, but specifically this is a 5' NTP. Because if we look at our numbering, this is 1', 2', and we know that this is 2'. It's a good kind of practice exercise to say, oh, well, there's my OH at 2', so I'm talking about ribose, 3', 4', and then this right here is 5'. Could you give me a Y or an N in the chat if that makes sense? Great. Okay. So it could be that phosphate. Where do we generally start off the numbering? That's a great question. Whenever we are looking at a nucleotide, or heck, this molecule right here, this brings back some of our terminology when it comes to sugars. This sugar is ribose. This is carbon number 1. That is our anomeric carbon. That anomeric carbon has a modification. That modification is that nitrogenous base. So we're going to start right there, but we're going to start and know that as our 1' carbon. So I would, I'm going to adjust your question a little bit. And it's not generally, this is always. So where the nitrogenous base is bound to your sugar, that is the 1' carbon. And 2', 3', 4', and 5'. Okay, awesome. So your phosphates are either going to be found on the C3' or the C5', in which case we're talking about a 5' or a 3' nucleotide. So look at where that phosphate is, and that's going to give you knowledge and information to designate it as a 3' or a 5' nucleotide. Now, here are some examples of, well, everything kind of culminating and being put together. These are ribo and deoxy ribonucleotides. So if we look at the top here, okay, if we look at the top here, and I'm going to open up, I've got 3 poll questions that I'm popping up right here, and just do these as we're going through this. If you look at the top, a fair question, and this is the sort of thing I want you to be able to answer and look at. If I showed you this molecule and I asked you, oh, well, is this DNA or RNA? You have a very quick way of answering that. And your quick way of answering that is not to look at the nitrogenous bases. Instead, look at, well, where is my carbon 2'? Oh, okay, here's 1', here is 2'. By the presence of that OH group on carbon 2', I know that these molecules on the top, or any molecules that have that OH group, are RNA molecules. Now, this first question is, on which carbon of the sugar is the nitrogenous base attached? Okay, well, here's my nitrogenous base, and boom, there's my 1' carbon. Yeah, so 1' carbon is the correct answer. Now, the other parts of this, the 5' carbon right here, well, that's going to attach to a phosphate group. That phosphate happens to be our gamma phosphate. Now, these at the top are our RNA molecules. That means that these down here are DNA molecules. Okay. Now, when it comes to symbols and names, the ones that you're going to hear most often are these: adenosine, guanosine, uridine, cytidine, deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine. One thing that I want you to recognize is if you're talking about a DNA molecule, you are going to say deoxy, then name that nitrogenous base. Now, the symbols, the best symbols for these are going to be Da and DAMP, DG, DGMP, DT, DTMP, DC, DCMP. Oh, but why are these MP? What does that mean? Well, that means monophosphate. Now, nucleotides do have specific names. For example, deoxyadenylate, deoxyguanylate, deoxythymidylate, deoxycytidylate. What is a more descriptive and maybe more comprehensive name would be something like this: deoxyadenosine. Okay, so deoxyadenosine, that is our nucleoside name. So we take nucleoside name, and this, what's in the parentheses here, is more comprehensive, and it's kind of the naming that I always like to use. The nucleoside name plus, if there's a phosphate there, which it's kind of implied here, plus 3 prime or 5 prime, okay, plus number of phosphates. So something like deoxyadenosine 3 prime triphosphate. Deoxyadenosine 3 prime. So deoxyadenosine tells me that I have a nucleoside. So I've got deoxyribose as my sugar. Deoxy gives me that. Adenosine tells me my nitrogenous base is an adenine. 3 prime tells me where my phosphate is. And then triphosphate tells me how many phosphates there are. Okay. So looking at all these questions, question one was, which carbon of the sugar is the nitrogenous base attached to? 1 prime carbon. 92% of the class answered that correctly. Question two, to distinguish a ribonucleotide from a deoxyribonucleotide, which carbon do we examine? Well, we examined the 2 prime carbon. Okay, 84% of the class got that right. And then the last question is, in the structure of a nucleotide, the 5 prime phosphate is attached to which part of the molecule? The 5 prime carbon of the sugar. So if it's telling you 5 prime phosphate, well, it's on the 5 prime carbon. Now I'm going to end that poll, go back, and now we've got some more some more info looking at these structures in a slightly different way. Um, this is kind of itemizing out adenine versus adenosine. Know the difference between adenine and adenosine. Adenine is the name of just the base. Adenosine is going to be the base plus the sugar. So I want you to know these two columns. That's it. The reason for that is you will typically hear terms like guanosine triphosphate. Well, that tells you that is an RNA molecule. Deoxyguanosine triphosphate tells you it's a DNA molecule with 3 phosphates on the 5 prime sugar or 5 prime carbon. Now, there are some different displays of monophosphates. So, adenosine 5 prime monophosphate, adenosine 3 prime monophosphate, adenosine 2 prime monophosphate, and then this last one down here, adenosine 2 prime 3 prime cyclic monophosphate. This right here, this cyclization can take place. More times than not, what's really important for biochemists to know, this one and this one. This is a little bit less common. Now, that this one being the 2' monophosphate. Now, this one in the lower right-hand corner, adenosine 2' 3' cyclic monophosphate. If you've taken any sort of cell biology class or maybe a physiology class, you've maybe heard about this stuff, CAMP. People sometimes just refer to it as CAMP. This is cyclic adenosine monophosphate. This is this structure right here is an example of that. This is what's known commonly as a secondary messenger. So what that means is that if a cell perceives some sort of hormone or stimuli, cyclic AMP is going to be produced to sort of amplify that signal. You know, the way that your body responds to caffeine or insulin is going to lead to the production of cyclic AMP. Now, I'm just showing you this as more or less an illustration of these different monophosphates. Now, there are also places in which you'll find an ADP molecule or a adenosine diphosphate as a part of a larger molecule. Now, we talked about nicotinamide adenine dinucleotide. Does anyone remember the type of enzymes or the class of enzymes that used NAD plus or NADH? I'll give you a hint. This one has a positive charge, this one's neutral. How can we get something to go from positive to neutral? It can gain an electron, right? But what I'm asking is what class of enzymes use this NAD plus? Oxidoreductases. Perfect. So these are cofactors. So an oxidoreductase, for example, will use a compound like this. Is that directly related to transfer of genetic material? No, it's not. It's directly related to transfer of energy, but not genetic information. So I want you to think about the molecules that build up DNA and RNA aren't exclusively for heritability or transfer of genetic information. Instead, they are molecules and compounds that carry energy. Okay, now when we think about DNA and RNA, it's pretty common to think about, well, a double-stranded piece of DNA or a single-stranded piece of RNA. And I mentioned those two because those are kind of the most common ways in which, and the classical ways in which, DNA and RNA are discussed. RNA as a single-stranded molecule and DNA as a double-stranded molecule. Now, when it comes to the primary structure of a DNA molecule, you're going to look at and see something like this, where we have a directionality. DNA reads from the 5' to the 3' direction. That's how we read it. The primary structure, we've got polymers of this molecule linking the 3' carbon of one sugar to the 5' carbon of an adjacent sugar. This type of linkage right here is known as a phosphodiester linkage. I always like to deconstruct that and say, okay, what's at the heart there? Phos, okay, phosphorus, diester, one ester, two esters. Done. We've got a phosphodiester linkage. Now, ribonucleic acid and deoxyribonucleic acid are the two terms that we use to describe these large polymer molecules. Now, another thing that I'd like you to think about with respect to that is a DNA molecule is known to have an overall negative charge. The reason for that is this phosphodiester linkage. Now, we'll get more into DNA's larger structure in just a moment, but that's our direction. 5' to 3' is the way that we read it. Phosphodiester linkages connect 3' carbons to 5' carbons. The sequence is always read. The convention is to always read it in 5' to 3'. So this DNA molecule right here, we would read the sequence as ATG. This RNA molecule, UGC. In terms of genetic information, this corresponds to or translates to N' or N termini to C termini that we see in proteins. So if you're thinking about, okay, well, I need to remember directionality of proteins, N to C. That's how we always read it. The primary structure tells us the N terminal amino acid all the way to the C terminal amino acid. DNA tells us 5' end to 3' end. That's all she wrote. Now, if we look at this right here, can anyone tell me, is this an what you're looking at here, is this an RNA or a DNA molecule, the one on the left here? Remember the clue that we have? Absolutely. This is an RNA molecule. We know it's RNA because what you guys looked for, there's two clues, two big clues. One is uracil. That's a one clue, looking at the nitrogenous base. Another is looking at that 2' OH group. Now, if you think, oh, well, I looked at this over this, great. You solved the problem. You got it right. Now, we've got an RNA molecule. RNA molecules are typically going to be single-stranded. DNA molecules are typically going to be double-stranded. Now, when it comes to naming or kind of looking at the sequence of one of these, it's pretty common to look at something like this, and look at something like this, but at the same time look at AUCG. Okay, these three sequences are all giving us the primary sequence of this molecule. Now, I want to draw your attention to a couple of things on it, so. Okay. When it comes to labeling and writing out the primary structure of a molecule, we've got version 1, version 2, and version 3. If I were to ask you the question, which of these is kind of the most comprehensive and provides the most information without information overload or information overkill? My answer to that question is 2. Now, a fair question for that is, well, answer option 1 has more information. So why is it not 1? And my answer to that is, well, we're talking about an RNA or a DNA molecule. What types of bonds connect every single sugar in an RNA or DNA molecule? For something to be an RNA or a DNA molecule, what type of bond? Okay, Melanie, your question is getting exactly where I'm going. In order for an RNA or DNA molecule to exist, you have to have a phosphodiester linkage. These P's that Melanie points out are the P's for the phosphates. These P's represent these phosphodiester linkages. Okay. Now, if those P's, if those phosphodiester linkages were not there, we wouldn't have a polymer of DNA or RNA. Could you give me a Y in the chat if you understand what I'm saying there? So putting those P's there to me is just kind of overkill. It's like, well, yeah, of course. Now, with that in mind, though, now let's look at answer options 2 and 3. Why is 2 better than 3? Is 2 better than 3? To answer that question, I want to look at the overall structure. I've got on the 5' end a phosphate. On the 3' end, I don't have a phosphate. This P right here indicates that my molecule has a phosphate on the 5' end. Now, my progression there is, could you synthesize a molecule without a 5' phosphate? Yes, you could. So what would that look like? Instead of this phosphate group, you would just have an OH. Now, this notation model right here, number 2, is more explicit. And it says, well, yeah, there is a 5' phosphate. If you had something like this, PAUCG, lowercase p, what does that tell me? Both of those phosphate groups, are they descriptive? Yeah, they are. What do they tell me? Where do I have phosphates on my molecule? I have phosphodiester linkages, but I know that. Where else do I have phosphates? Got one on the 5' end. Asthma. Perfect. 5 prime end and 3 prime end. So that's why I like this. And that's one of the things that I want you to recognize. If you see a lowercase p in front, I want you to recognize that, oh, okay, well, I'm reading my molecule 5 prime to 3 prime. That says that I have a 5 prime phosphate group there. Okay. Now, all right, good. So now we've got a couple more questions that I want to ask you guys, and I'm going to open these up and just kind of keep them up for the next few slides and answer them as you see, as you're ready to. So here is our double helical structure, our secondary structure of a DNA molecule. Major parts or geographical structures are the major groove and the minor groove. These are regions of a DNA molecule that allow for proteins to interact with them. Now, the sugar phosphate backbone, well, that's all your phosphodiester linkages and your sugars. If I jump back, this is what makes up your backbone. Those sugar phosphate, um, that sugar phosphate backbone is on the outside of a DNA double helix. What that means then is that your nitrogenous bases are in the middle of that DNA structure. Now, this sugar phosphate backbone gives the negative charge. So if you ever talk about a DNA molecule having a negative or a positive charge, or what is the charge of a DNA molecule? Well, the sugar phosphate backbone gives it an overall negative charge. Now, the backbone, you have two strands that are anti-parallel of one another. So they run opposite of one another. Your major and minor grooves are the interactions, or sorry, the regions in which proteins can interact with this overall structure. Now, if we dissect this and kind of, instead of, if we flatten out the double helix, it looks a little bit like this on the left-hand side here. And this shows us the interactions between our nitrogenous bases. G pairs up with C, A pairs up with T. You've got all of these negative charges right here. Everywhere I'm drawing a negative, you know what that represents? That represents a phosphate. Now, this is, keep in mind, a flattened out double helix. What's important to look at with this double helix is that what are the nitrogenous bases that interact with one another? Well, G and C interact with one another. Could you tell me in the chat how many bonds exist between G and C? 3. That's important. Compare that to A and T. There's 2 between A and T. Now, with that in mind, that's a constant. 3 bonds, 3 hydrogen bonds, 1, 2, and 3, form the interaction between G and C, guanine and cytosine, and 2 form the interaction between A and T. Now, what do you think is stronger? 3 hydrogen bonds or 2 hydrogen bonds? 3. Now, is that significant? It is. That's significant because, well, what that means is that if you have a molecule or if you have an organism that has a high GC content genome, what does that mean? Well, that means it has a high percentage of guanine and cytosine within its genome. Now, with that in mind, A and T, G and C, those are all of your base pairs. G is not going to pair up with T, and C is not going to pair up with A. So if I were to say, and this is going to what's known as Chargaff's rule, let's say that I told you that you had 28% of a genome that was G. You might say, "Oh, cool, thanks. Thanks for sharing that information. That's delightful." Um, is there anything else that could you fill in the gaps there? Is there any other information that you'd be able to say, "Oh, well, based on that, I know something else. 28% of the genome is G. What percentage of the genome is C? Yeah, it's 28% C. Okay, so that would in effect mean that the genome is 56% GC. Does everyone agree with that? Give me a Y or an N to the chat. Okay. Then arguably that means that 44% of your genome is AT, and that also means that 22% is A, 22% is T. Could you give me a one in the chat if that makes sense? If all those numbers add up to you. Okay. Now, from a genome standpoint, if you had two different organisms and one of them had a high AT content, one of them had a high GC content, the conditions in which they live are going to be different. Now, GC content is not going to vary from one person to the next, but certain bacteria, certain organisms that live in the bottom of the ocean, they're going to have different GC contents. And that all boils down to, well, how many hydrogen bonds are holding these molecules together? Okay, so this is what a DNA molecule is going to look like. Here's the number of base pairs that are going to hold it together. Now, an RNA molecule, RNA can get kind of hairy. RNA can get kind of crazy and cool, though, because an RNA molecule, just as this is sort of your secondary structure in structures that a RNA molecule can take on. You can have a helical region. So this is just a hypothetical RNA molecule denoted by the U's present. Um, and RNA is typically going to appear as a single-stranded molecule, but could anyone tell me what type of bonds do the nitrogenous bases like to find? Nitrogenous bases will form blank bonds. So between two nitrogenous bases, A and T, what type of bond is formed between A and T? Tell me in the chat. And you could just go with a single letter of it, or you can type the whole word. Hydrogen. Perfect. Hydrogen bond. Okay. Now, what type of bonding do water molecules like to form? Yeah, water molecules love to hydrogen bond. So if you had a nitrogenous base that liked to hydrogen bond and water that liked to hydrogen bond, um, that's kind of a match made in heaven. Hydrogen bonding. So nitrogenous bases will form hydrogen bonds. Now, what I want to clarify though is they'll form hydrogen bonds with anything else that will form hydrogen bonds. Give me a one if that makes sense to you. So what that means is that could be another nitrogenous base, that could be an H2O molecule, that could be an alcohol functional group, that could be a any sort of amine. So my point is that an RNA molecule is typically going to be, it's classically described as a single-stranded molecule, but because of its love of hydrogen bonding, it will form hydrogen bonds with other parts of itself, as well as other parts of the environment. Give me a five if that makes sense to you. So kind of a consequence of that is we'll have these kind of crazy structures form. A single-stranded RNA molecule, well, nitrogenous bases are going to pair up and you're going to lead to this helix or this helix, a different helix, helices, loops, bulges, hairpin loops. All of these things are going to form based on where hydrogen bonds form. Now, I don't want you to stress about, oh, well, what are the specific structures? I don't need you to know that. I just want you to kind of think about an RNA molecule as because it doesn't have the double helix structure that DNA does, what that means is it's going to hydrogen bond with lots of stuff. And when it hydrogen bonds with lots of stuff, that can lead to an RNA molecule being unstable. And where I'm going with that is, well, if I'm just gonna actually, here we go. Um, if we have a single-stranded RNA molecule, here's that single-stranded RNA molecule. If I have this region, that's going to hydrogen bond with an amine. It's going to hydrogen bond with that. And then this portion of it's going to hydrogen bond with an H2O somewhere else. There's stress and strain that's being applied to that molecule. And those could be going kind of in opposite directions. Could you give me a 9 in the chat if, okay, I follow that. Okay, great. So that's what I want you to know about RNA molecules. It's important to know the what's known as Chargaff's rule or Chargaff's rule, the base pairing that exists. A pairs with T, G pairs with C, how that leads to different percentages and the implications of that downstream. Now, with that, I'm going to go ahead and transition to the next slide deck, which I added this to the chat. I'm going to go ahead and grab it again, though. I'm going to copy link and add this slide deck, and I'm going to close this one out. So end slideshow, keep annotations. And now we're going to, this one is titled DNA Technologies. So I copied the link, or techniques, sorry, techniques. Now I need to go back. Okay. So we're kind of digging deeper into DNA. And if you're familiar with the polymerase chain reaction or any way in which you can manipulate DNA, that's going to be good context and foundational information for you for this. So first and foremost, DNA can be manipulated, or nucleic acids can be manipulated. Nucleic acid fragments are generally easy to work with since they can be sequenced, duplicated, modified, and expressed. These first three are, well, you can purify that molecule and analyze it. This last one is you can take a DNA molecule and make it into a protein with E. coli or yeast or something like that. And the word that's typically used for this is a recombinant protein. If you ever buy a dairy product and you see that that dairy product says never RBGH, that little R stands for recombinant. And what that means is that wherever that dairy product came from, there was never any recombinant bovine growth hormone used. So that cow that grew, well, it grew and it gave milk how it naturally would. With that said, dairy industry is kind of crazy. Um, is the expression like making cDNA from RNA? That's a very, yes, absolutely. So you'd take, in order to do this, you're going to take a DNA, your chances are you're going to isolate. Okay. The process, and this is a little bit of an aside. This is not something I'm going to test you over for the nuances of expression and the details of expression, but instead what you're going to do is you're going to isolate RNA. And RNA comes in a couple of different forms. You're going to isolate mRNA, messenger RNA. That messenger RNA, you're going to use reverse transcriptase, an enzyme that takes an RNA molecule to make DNA. And with that reverse transcriptase, you're going to make a reverse transcript, and that's going to allow you to make a cDNA of that gene that you're interested in. That cDNA, you're going to be able to put that into a plasmid or a vector and put that into something like E. coli, and then you're going to be able to overexpress that protein and get a lot of that protein. So that actually kind of cycles back to when we were talking about why do we not sequence proteins very much? Well, the reason is because most of the time, whenever we're analyzing a protein, we're working with the RNA. We're not going to try to purify a protein and say, oh, what is this? Because it's technically much more demanding. Okay, now that's my conclusion of the aside. I hope that I answered your question, Dylan. I hope that I gave you supplemental information that was either affirmational or, hey, I didn't know that. Um, so many times it is easier to get protein sequence information from an organism's DNA than it is from purified protein. If you think about genome analysis versus proteome analysis, genome analysis is so much easier. Sequence a genome, you can do that 23andMe. Just as in protein sequencing, we must cleave this DNA into specific fragments so that we can put a piece of DNA into a plasmid. We must sequence each fragment and put those fragments back in the correct order. So, this thing right here, sequencing of DNA, back in the 70s, if you sequenced the gene, 1970s, if you sequenced the gene, awesome. You probably just earned yourself a PhD. And the reason for that is because the technical challenges, um, the work that had to be done, the technology that was available. But nowadays, if you sequence a gene, I'll say 2025, if you sequence a gene and it takes you, or to sequence an entire gene, you should be able to do the work and submit that. You can, well, let me rephrase that. You can get a DNA sequence back within, say, 24 hours, just because the sequencing technology is so accessible and easy to use nowadays. Okay, so DNA or nucleic acid manipulations, more times than not, people are analyzing DNA because DNA is so much more stable. Um, RNA is very sensitive because, like I said, it can hydrogen bond to everything. So it will hydrogen bond and just kind of get obliterated. In addition to that, there's RNases everywhere. So my experience working with RNA as a graduate student did not go well because I was kind of reckless with my sequence or working with it because you're supposed to like sterilize your entire bench, and I would do things like, yeah, that looks good enough, and lo and behold, my DNA would all get degraded. Now, where we're going to go from this is the cleaving into specific fragments. So we're using molecular scissors, but rather than trypsin, chymotrypsin, and elastase, which cut peptide bonds, we are going to be looking at restriction enzymes that cut either hydrogen bonds or what's the other type of bond that hold a DNA molecule together? Add it to the chat. Now, spoiler alert, it's not going to be hydrogen bonds. So restriction enzymes don't do anything to hydrogen bonds. Phosphodiester bonds, perfect. So restriction enzymes love to cut up phosphodiester bonds. So these cleave double-stranded DNA. Sometimes you'll see this written as dsDNA, and I'll even sometimes use dsDNA. These are purified from bacteria. This is an important piece of information I'm going to bring back in just a little bit. Now, there are three types of restriction enzymes. Type 1, and you can probably guess there's type 2 and type 3. Type 1 requires ATP, and they offer non-specific cleavage. What that means is they are going to see a phosphodiester bond, and all the phosphodiester bonds, they're going to say, cut, 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 cut. Type 2 restriction enzymes, they do not require ATP, but they're very specific. What that means is that they're going to look for a specific phosphodiester bond and cut that. Now, I'll get to the how is it specific versus not specific in just a moment. Type 3 are going to require ATP, and they are also specific. Now, what we are going to focus in on is type 2 restriction enzymes. These are sometimes referred to as restriction endonucleases. Keep in mind, we're cutting a double-stranded piece of DNA. So what that means is that the two strands are reverse complements of one another. Now here is a series of names. I want you to focus in on this column here on the left, the enzyme name. And I'm going to bring your attention down to these two right here. Eco. Okay, Eco. Whenever I hear that word, I think of a Sega Genesis game called Echo the Dolphin. Very boring game. I also think of ecotype. I think of ecology. But then if I separate this out and I say ECO, what comes to mind whenever I say E, maybe throw a period there, CO? add to the chat what you think about, especially in the context of bacteria. E. coli, absolutely. These two restriction enzymes are examples that came from Escherichia coli, E. coli. I love to say Escherichia because for the longest time I struggled to say that word. Just say like Escherichia, or I'd just get it wrong altogether and I'd say yeast? No, yeast is a fungus and it is, well, it's not bacteria. Um, at any rate, it's eukaryote too. Okay, so ECO R1, ECO R2. So these are two restriction enzymes that were purified and isolated from bacteria. Now, I want you to focus in on ECO R1. What ECO R1 is going to do is it's going to see a double-stranded piece of DNA, and it's going to see G, A, A, T, T, C. And it's going to see the phosphodiester linkage between G and A right here. And it's going to cut there. Okay, so that's going to cut one strand. Could you give me a 1 in the chat if you follow that? Okay. Now, the other strand. Both strands are 5' to 3'. So this is 5' to 3'. Well, this is a G complements the C, A complements a T, A complements a T, T complements an A, T complements an A, and C complements a G. So it's going to see, oh, it's going to cut right there. Is this, and this isn't intended to be a trick question, or no, it's not intended to be a trick statement, but this EcoR1, it's scanning this strand, it's going to cut between G and A, and then it's going to scan the other strand. Where is it going to cut? Well, it's also cutting between G and A. So it's going to cut those phosphodiester bonds. Now, did EcoR1 here break any hydrogen bonds? Nope. The hydrogen bonds here, 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 they're there. EcoR1 doesn't do anything with this. But most importantly, it is breaking phosphodiester linkages. Now, if you think about the kind of strength and security of bonds, I'm going to go over here. Well, this is another illustration, kind of what I was showing you, but a little bit different. A phosphodiester bond is a covalent bond, strong. Can it cut there specifically because it goes in order 5' to 3' for the GA? Yes, Melanie, spot on. Great observation, great question. Thank you so much. It looks for that GA, but specifically it's looking for GAATTC. So it's looking for all 6 of those nitrogenous bases in the sequence of them. Now, thinking about a double-stranded piece of DNA, this restriction enzyme's kind of scanning in and it's like, hey, I can see in those major and minor grooves, I can see the nitrogenous bases that are there. So I'm going to cut between G and A as long as it's GAATTC. I'm specifically looking for that. So this is a type 2, does not require ATP, and it specifically cuts at this specific location. Could you give me a 9 in the chat? A 9 star, if you're good with that, and that makes sense. Okay, great. Now, here's the thing. This is a recognition sequence. So EcoR1 says GAATTC. Got it. I'm going to cut between the G and A because I found GAATTC. If EcoR1 sees GAATTG, what do you think it's going to do? Is it going to cut or just say, nope, not what I'm looking for? C for cut or N for nope, not what I'm looking for. Perfect. It does not care about that. It's excited, but then when it sees that G, it's like, no, not me. Okay. Now, what is going to happen here is that there are hydrogen bonds holding these nitrogenous bases together. The covalent bonds are super strong. Hydrogen bonds, by comparison, are kind of weak. In huge numbers, hydrogen bonds collectively bring a lot of energy and they are stable. But what's going to happen is that these hydrogen bonds here, 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 they're all going to be disrupted enough that they can't hold this molecule together. So that is going to result in a separation that looks a little bit like this and this. These are commonly known as sticky ends. The way that I always kind of think about it is, and you can do this just as well, but look at my index finger and my ring finger. Ring finger, or sorry, index finger and middle finger. I'm trying to avoid talking about middle fingers because I have a 5-year-old who's always like, I'm holding my middle finger up. And then he drops the ring finger and the index finger, and then he's just like flicking everyone off. It's a struggle. So I've got my middle finger and my index finger. Think about the junction between the tip of my index finger and the tip of my middle finger as the phosphodiester linkage that gets cut. Okay. Sure, these hydrogen bonds are holding my middle fingers together. Can everyone give me a minus in the chat if they follow that? But they're not strong enough to hold the whole apparatus together. So you clip those phosphodiester linkages here and here respectively, and then it's almost like, well, it's not strong enough to stay intact. Okay, so so the result whenever you have this is what's known as a 5' overhang. Here's a 5' region that overhangs and is going to look for hydrogen bonding pairs. These are also known as sticky ends. Now, if we look at this enzyme, EcoRV, EcoR5, this, well, there's no collateral damage of a hydrogen bond being disrupted. It's going to cut between those two nitrogenous bases, or that it's going to cut those phosphodiester linkages that are directly across from one another. The way I always kind of think about this is there's no 5' overhang, and these are blunt ends. Add to that sticky ends, blunt ends. The question is, or the question that I like to ask is, with sticky ends, H bonds are indirectly broken because they're not strong enough to keep the whole thing intact. These 4 hydrogen bond pairs are not strong enough to hold the molecule together. Blunt ends, by comparison, well, no H bonds are broken at all. Okay. Can anyone now, with that said, if we go back to this, I'm not interested in you knowing all of these sequences and all these recognition patterns. That's not the point. What is the point is to recognize that these recognition sequences are typically going to be palindromic. And what I mean by that, a palindrome is something that when read in one direction and in the opposite direction, it's the same. This is kind of, yeah. So if you just took the word madam, M-A-D-A-M, in one direction, it spells madam, and in the opposite direction, it spells madam. So there's that. Typically, your recognition sequences are going to be 4, 6. Oh, yeah, good. 4, 6, or more bases, which is what we see on this slide. Here's 4 base pairs. Then here's considerably longer. Now, if you look at something like this says GCC, and then N corresponds to any nitrogenous base. And it's just going to go on from there. Y corresponds to any pyrimidine, and then R stands for purine. So if I were to give you something like this and say, all right, well, we cut between the G and the A, is that going to result in sticky ends or blunt ends? Is there going to be a 5' overhang? Are there going to be hydrogen bonds broken at all? That's the sort of question that I would ask you. So I would give you a hypothetical pairing like this. So, sorry, I'd give you a picture like this right here, and say, okay, well, it cuts right here. Is that going to result in sticky ends or is it going to result in blunt ends? Now, I'm going to erase this arrow because I don't want you to look at this and be like, oh, but he drew an arrow between here. Nope, that was just a coincidence. Does everyone understand that? Give me a plus in the chat if you follow that. Okay, great. So now DNA gel electrophoresis, I've got some more slides that I want to, sorry, I got another set of questions here that I want to ask regarding restriction endonucleases. So feel free to answer those as you want and when you're ready. DNA gel electrophoresis, we've got right here. This is what DNA gel electrophoresis looks like. Your smaller bands run through the gel fastest. Now, one thing that's different from protein gel electrophoresis and DNA gel electrophoresis is you have to treat a protein with a certain chemical. Does anyone remember the chemical that we use to provide a protein with a uniform negative charge? Add it to the chat. I'll give you a hint. I'm looking for 3 letters. So when you are analyzing a protein by gel electrophoresis, you have to use that substance sodium dodecyl sulfate, SDS. Now, you don't have to do that with DNA. Why? Well, we've got a negative charge running towards a positive charge. What's the overall charge on a DNA molecule? That's right, it's negative. So we're good. We don't have to worry about that. Now, what is valuable to know about DNA gel electrophoresis is it is a technique that you can use to confirm something. For instance, this right here, this what looks like a gray strip almost, it looks like a piece of tape that somebody marked on with a Sharpie in 2 places. What this is showing is 2 bands that correspond to pieces of DNA that result from being cut with this molecule right here, with this restriction endonuclease, HIND3. Now, one thing that people can do is they can take multiple restriction enzymes and cut one piece of DNA with multiple restriction enzymes, and then they can map them. Now, if we look at this right here, okay, so this blue strip represents our linear piece of double-stranded DNA. This represents, this start is basically base pair 0, and then this right here is base pair 4000. So it's from nucleotide 1 to nucleotide 4000. Could you give me, does everyone understand that if you do press M in the chat, if you don't press W? Okay. So I'm just showing you that we flattened out this piece of DNA and we have no idea what the sequence is. But if we cut this piece of DNA with HIND3 and then we run the resulting digestion products in a DNA gel electrophoresis, we see that there's one band that's 2.8 kilobase pairs or 2,800 base pairs. There's another one that's 1,200 base pairs. Okay. Huh. Is that significant? Well, 2.8 plus 1.2, that adds up to 4. Good. That's 4,000. Now, but HIND3, do we know the recognition sequence that HIND3 has? Well, if we look back at this table, we can see that HIND3 cuts AAGCTT. Does everyone follow that and agree with that? Okay. Now, what that means, and this is what I said earlier about sequencing a piece of DNA or sequencing a gene would merit a PhD back in the 70s. This is, again, 4,000 base pairs. That's a lot. If you can identify where HIND3 cuts, You have learned something. You have learned that the sequence right at that location is AAGCTT. Okay. So if we were trying to figure out the sequence for this piece of DNA, previously we did not know any of the 4,000 base pairs. We did not know a single, we did not know the sequence at all. Does everyone agree with that? Add a seven in the chat if yeah, I agree with that. Or any other character if no, you don't follow. Okay. But you know what we just did? We cut this with Hind 3. Now all of a sudden, and I mean this kind of in jest, but also seriousness, this is back in the 1970s. Good news. We found out the sequence and the positioning of six of the nucleotides. We now only have 3,994 base pairs left to solve and identify their sequence. How's everyone feeling about this? This is great. Only 3,994. Okay. So then you'd cut it with another enzyme. You cut it with a different enzyme, and that's going to say, okay, well, when you cut this linear piece of DNA, you get these products. Okay, so now all of a sudden, oh man, we've got more nucleotide, more sequence that we can add. Does everyone follow that? Give me a Y or an N if they understand that. Okay. Then when you do what's known as a double digestion, you can do a little bit more piecing together. And now all of a sudden you can say, oh, well, whenever I reassemble this piece of DNA, well, I have to assemble this, this, this. This piece goes here, this piece goes here because of the gel electrophoresis results that I got. Okay, so I want to stress that as like, listen, this is how DNA sequencing used to go. Now, the sorts of questions I want you guys to be able to answer are if you have a linear or a circular piece of DNA. If you have a linear piece of DNA, that looks like this. A circular piece of DNA looks like this. And if you've ever been to a wedding, then there's always that moment when people are reciting the vows that they got off of Pinterest sometimes, and they say, a ring is circular and has no ending. You're right. Absolutely. Perfect. And so I give you this ring. Done. Um, if you cut a ring with a restriction enzyme that cuts it in one place, let's say it cuts right here, how many bands will show up on this DNA gel? Well, if you cut a circular piece of DNA and it cuts right there, you're going to get one band. Whereas a linear piece of DNA, if this restriction enzyme cuts directly in the middle, what are you going to get? How many bands are going to show up on your DNA gel? Two. Okay. So I want you to know that sort of thing. I want you to know the difference between that. Now, I'm going to jump ahead a little bit. That's kind of the point of that. And what we're going to do is we're going to skip over this restriction fragment size and this restriction fragment length polymorphisms, also known as RIFLIPs. And we're going to go on to this technique known as the polymerase chain reaction. Give me a one in the chat if you have heard of the polymerase chain reaction prior to this class. And give me a five in the chat if you've done the polymerase chain reaction. Now, we had the experience of a global pandemic where now all of a sudden everyone's well versed in what oh, you got a PCR test? Oh, okay, that's more sensitive. Yep, yep, got that. That's what we're talking about. The polymerase chain reaction was utilized. So the polymerase chain reaction was developed in started in the 70s, but went on to the 80s. And I believe it was in 1993. Dr. Kary Mullis was one of two people, maybe three people who was awarded the Nobel Prize for it. If you've ever heard of Kary Mullis, he is a character to say the least. One of the things that he credits his kind of inception and development of the polymerase chain reaction is psychedelic drugs. Um, and he hallucinated part of like theory on how to do it. That's always the story that I've heard. Specifically, I heard it was like a green raccoon. I don't know if the green raccoon told him how to do it, in which case, Dr. Mullis, do you really deserve the Nobel Prize, or does this mysterious green raccoon really deserve the Nobel Prize? At any rate, the point is the polymerase chain reaction is a technique that completely changed life science research. It's, it was kind of commercialized and blew up back in the 80s and 90s. It's, I don't know if this isn't too reckless of an opinion, but I kind of think that it had the same effect on life science research and science research as isolation and determination of the double helix had. Now, what this allows you to do, what this technique allows you to do is take one double-stranded piece of DNA and make almost an infinite number of copies of that one piece of DNA very quickly. Now, the polymerase chain reaction consists of basically two cycles. Now, these steps or cycles are repeated kind of indefinitely, maybe not indefinitely, but a certain number of times. Usually that is between 30 and 35. Now, the requirements for the polymerase chain reaction are you have to have, well, an original template, a piece of DNA that you want to synthesize. You want to synthesize and replicate. Then you have to have what are known as primers. These are oligonucleotide primers. Oligonucleotides, they're typically 15 to maybe 40 base pairs long, and they are going to directly complement or reverse complement each of your two strands of DNA. So you have this double-stranded piece of DNA, and then you have these primers that are reverse complements of short regions of each one of those. You also have a heat-stable DNA polymerase. If you've ever heard of DNA polymerase, it is an enzyme that's, well, it's capable of making a polymer of DNA. A heat-stable DNA polymerase is important for PCR because in PCR you go through a couple of different temperature fluctuations. You also have to have four DNTPs or deoxynucleotide triphosphates as opposed to nucleotide triphosphates. Then you've also got to have magnesium 2 plus. Magnesium is important. It's a divalent cation of a certain size that really helps your heat-stable DNA polymerase work. Now, the steps are as follows. I'm going to highlight this. The first step is to heat up your double-stranded piece of DNA. Usually this takes place in a matter of, 10 to 15 seconds at about 95 to 98 degrees Celsius. The next step is right here where you've separated the pieces of DNA, but then you allow your oligonucleotide primers to anneal. So sometimes this first step is called denaturation, or it's sometimes called melting to separate, or it's even called separation. The next step is your hybridization, where your oligonucleotide primers anneal or bind to your template strands. Then the last step is your polymerization. Okay, so what this is illustrating right here and right here is the hybridization. Your oligonucleotide primers bind to the two template strands. Your polymerization is where the enzyme DNA polymerase jumps onto this region right here and right here, respectively. DNA polymerase, what it is looking for a molecule that is partially double-stranded, but then partially single-stranded. Whenever it sees that partially single-stranded region, it says, oh, okay, well, I'm going to use DNTPs, and I'm going to synthesize and complete this molecule. So that is going to be repeated over and over and over again. Usually, it's about 30 to 35x. Our temperatures are 95 to 98 for that initial denaturation. I'm just going to give you a simple 95 degrees. The hybridization, well, that's taking place at about 55 degrees, and the polymerization is happening at 72 degrees. So you will go through, when you're doing PCR, you're going to heat the components of your mixture up to 95, then cool them down to 55, then 72, 95, 55, 72, 95, 55, 72, 95, 55, 72, and so on. You're going to do that about 30 times. Now, that is why you have to have a heat stable DNA polymerase. If you were to, say, take some of any number of my cells, Joel cells, and isolate DNA polymerase, I've got DNA polymerase in there. It's great. It would not be able to resist or it would not be able to continue functioning based on those temperature cycles. If you heated it up to 95, that DNA polymerase is toast. So you had to go to an extremophilic bacteria or an extremophilic microorganism. Um, Now, with that said, there are tons of different applications of PCR. Forensics, you've got DNA fingerprinting, you've got comparison of um, traits, you've got comparison of different pieces of DNA, detection of infectious bacteria and viruses. In addition to that, this is the foundation for DNA sequencing. The type of DNA sequencing that's done on a contemporary level is going to be what's known as a chain terminator procedure. And you know what? I'm going to share a couple of poll questions. Let me. This is good. Okay. Um, I'm gonna launch. I've got four poll questions that I want you to answer. Um, I'll talk about DNA sequencing, but I'm not going to ask any questions about DNA sequencing today. We'll jump off of this on Tuesday for a little bit. It looks like we'll do some stuff with lipids, and that will be on the exam, but not a huge amount of stuff regarding lipids will be on this exam. I'll give you an update of that later today. Or not today, I'm sorry. After I've revised the exam to reflect what we actually covered. But DNA sequencing. Very similar to PCR. The difference is you use a fragment, a piece of a DNA polymerase known as the Kleinow fragment, sometimes pronounced Kleinow fragment, Kleinow fragment, whatever. You have to do or you have to use a single strand of DNA and a DNA primer. So you're not using both strands. They'll both be in there, but you're only going to have a primer that complements one of them. You're going to use nucleoside triphosphates. Okay, cool. Just like in PCR. But then you're also going to use small amount of, oh man, DDNTP, dideoxynucleotide triphosphate. Anyone in the chat, this lowercase d right here that I've just circled, what carbon does that correspond to? Well, this lowercase d is referencing our 2 prime. Yeah. Now, 3 prime. Oh, wait. Could anyone tell me when we talk about a phosphodiester linkage, which carbons are connected through a phosphodiester linkage between we got two different sugars. What are the carbons that are linked there? 5. But I want to clarify, I'm not seeing prime. It is important to say 5 prime and 3 prime. So yeah, our 5 prime and 3 prime. In order for that phosphodiester linkage to exist, you have to have a 3 prime OH group. What if you don't have a 3 prime OH group? Are you going to be able to make a phosphodiester linkage between two sugars? No, not at all. And that's what's being exploited when you're doing DNA sequencing. So here what you have is this DDNTP analog. You don't have an OH on that 3 prime or the 2 prime. So what's going to happen is you've got your primer, great. You're going to bring in a deoxynucleotide triphosphate, and you're going to be adding more and more base pairs until you hit a DDNTP. And then you're going to add that DDNTP, and that's going to stop the reaction right there. Now, like I said, I'm going to get more into this on Tuesday, In the meantime, though, I hope you guys have a great weekend. Thank you very much for participating, answering all my questions. Just as a little bit of a review, the questions that were our closers, which of the following correctly describes the orientation of DNA strands? One strand runs 5' and the other runs 3'. If you're looking at them directly across from one another, that is true. But they're reverse complements of one another also. So I was looking for one strand runs 5' and the other is 3' because we're looking at them kind of together. Um, which of the following is a deoxyribonucleotide containing adenine? Okay, all of these except for, well, DCMP, I feel like that was someone trolling me. Understood. Respect. AMP, ATP, and DAMP, they all contain adenine, but DAMP is the only one that has the deoxy adenine. Three, a molecule composed of ribose, guanine. Okay, so we've got no phosphate group. That is known as guanosine. That is a uh, ribonucleoside. Then, last but not least, so guanosine is the correct answer there. Last but not least, question 4. What feature of PCR allows it to only the targeted DNA region to be amplified? Heat stable polymerase? No. Fluorescent DNTPs? Nope. Sequence specific primers? Yes. The sequence specific primers are going to align with one region of the molecule, and that's where your DNA polymerase is going to jump on and synthesize more and more. Okay, everyone. I'm sorry, I didn't realize there were 4 questions. Don't worry about it. It's okay. I hope that you guys had a good day. I hope that you learned some stuff. And I will see you on Tuesday. If there's anything that comes up over the course of the weekend, please let me know. I'm going to send an updated kind of guide for preparing for the exam. I'm going to get writing the exam tomorrow or revising the exam tomorrow. So I'll have a better idea of that and be able to cover some things that we will cover on Tuesday. Just to give you. So, okay. Absolutely. Thank you. I'm going to stop my recording.